welcome to the Endpoints podcast from the ALS Therapy Development Institute. Today I'm joined by Dr. Fernando Vieira, Chief Scientific Officer at ALS TDI, and we're going to talk about a paper recently published by ALS TDI scientists in Nature's Scientific Reports. In this paper, our scientists indicate that protein aggregates are protective against motor neuron disease progression caused by SOD1 mutations. So Fernando, there's a lot to discuss within this, but let's start with what are protein aggregates?
So protein aggregates, one way to describe them is they're protein clumps. So thinking about a way that might make sense. So when you take, say, table salt and put it into a glass of water, it disappears, right? It gets solubilized. And as the water evaporates, or if you add too much salt, you start to see crystals form. So it kind of clumps, and then you're able to visually see it. Proteins in a cell are typically in a solution. So it's something kind of floating around, and you couldn't see it with the naked eye. It's too small. There's only like one or two of these molecules floating around. When you have a protein aggregate, you essentially have kind of a clump of proteins that forms within or outside of a cell that's actually now large enough for you to see with like a light microscope, or in some cases even your naked eye. So that's a protein aggregate.
Okay. So in the abstract it reads, we found that the central nervous system regions least affected by disease had the most aggregated SOD1 protein. Why is this interesting?
It's interesting because ALS, along with other neurodegenerative diseases, including Alzheimer's, Parkinson's, Huntington's disease, and others, they sort of all manifest with these protein aggregates. So these clumps of proteins that pile up in the central nervous system, so in the brain or the spinal cord. And at least for me, when I was getting into the field of ALS a while ago, it became really easy to assume that, well, neurodegenerative diseases are bad. They all have these protein aggregates, ergo, protein aggregates are bad, and that they would cause neurons to die. And it's sort of, that idea is further bolstered by the fact that the protein clumps are typically made up of, or at least some of their constituent parts, are the proteins encoded by the genes that are mutated that cause the disease, right? So there are a lot of reasons that point you to think protein aggregates are bad. But we actually found a few different data points that suggested differences here. Number one, what you just alluded to, is the fact that the parts of the spinal cord in these mice that we studied that had the highest aggregate load are the parts that suffer from the least motor neuron degeneration and have the fewest ALS-related symptoms associated with them. Whereas the lumbar spinal cord, the part that's most affected, where most of the motor neurons die and where you see severe paralysis in the hind limbs, that actually had the least aggregated protein. So it kind of turned the paradigm on its head to us.
Okay, so we come into the lab every day and we see all the familiar faces. So who in the lab worked on this particular paper and project?
So a bunch of people worked on it. Everybody who works on the project in the animal care facility, but specifically, a ton of the experiments in the lab were run by Cindy Gill and James Phelan. And they worked really hard, number one, to develop the assay system. So what that means is the systems that allow you to measure these different types of SOD1 really, really specifically. And then once they developed the systems, then they just had to execute over and over and over again a lot of experiments to get high quality data. So it was a huge effort over a long time by really great scientists.
And just for context, how long have we been working on this in our lab here?
Probably about 5 years in one way, shape, or form. It started out of a collaboration with a company called Neuroimmune. We actually published a companion paper sort of in Science Translational Medicine in December with Neuroimmune, where we were exploring an antibody reagent that could really, really do a good job of detecting misfolded SOD1. And so we explored it a couple different ways. Number one, to try to understand the behavior of SOD1 and misfolded SOD1 driving disease. And that's a lot of what this paper talks about. But also an antibody against misfolded SOD1 as a therapeutic. So if you can target this misfolded protein and either clear it or activate the immune system to respond to it, maybe you'd have a therapeutic effect. This work is a part of a bigger project with Neuroimmune around all of these types of reagents.
Okay. We're mentioning SOD1 kind of a lot here. We know that SOD1 mutation makes up for about 2% of all ALS cases. Mm-hmm. Is there a way that this could influence greater ALS research?
So, yes. So, a few different ways. Some people believe that misfolded SOD1 is playing a role in more cases of ALS, so even sporadic ALS. In fact, our collaborators at Neuroimmune have shown some evidence in support of that, but that's a really controversial point. But more generally, the concept that we talk about in this paper, where we're talking about protein aggregation, it does have, we're not sure, right? Ultimately, we're not certain that it will apply to all ALS or other neurodegenerative diseases. But the idea that the aggregates are protective, that the protein aggregate might actually be kind of sopping up or sequestering damaging misfolded SOD1 and putting it in a place in the cell that's not problematic, that could have broader implications. And if you're a person or a researcher who's working on or thinking about proteinopathies, so diseases that involve protein misfolding, this is definitely a consideration that you would want to think about when you're working with A beta for Alzheimer's or Huntington and Huntington's disease.
Okay. And our researchers made these discoveries in mice. So how do you think this discovery will sort of influence drug development for humans?
So there are, at least for us, right? So we are a drug discovery and drug development institute. There are so many ideas that we're always pursuing. There's certainly not a dearth of good ideas out there. And so you do need to find ways to prioritize the projects that you're going to work on. And among the good ideas that people work on are approaches to break up aggregates, right? So therapeutics designed to bust them up. And for us, I think these data deprioritized those therapeutic approaches. We're not going to fully, ignore them because you always need to integrate new information into your model. But right now, breaking up aggregates is not a high priority. In fact, maybe it would be interesting to look at therapeutic approaches that actually potentiate aggregation, that could actually help a cell do a good job of pulling those misfolded proteins into an aggregate.
Okay, that was really interesting. So thanks for joining today, Fernando, and for giving us some new information about this paper. If anyone has any questions or comments about this paper or the Endpoints podcast, feel free to email us at endpoints@als.net. Thank you for listening.
Thank you.